Yes, okay. Welcome everyone to a new episode of the webinar series Unraveling T-cell Recognition Insights from Immunology and AI. My name is Hashem Kui, and I am delighted to be hosting this series where we invite researchers and innovators from around the world to share their work with us. This series is held mostly first Monday of each week at 1300 UK time. The announcements are usually made either via my LinkedIn page or dedicated mailing list. So please subscribe to make sure that you are tuned and updated with this announcement. Please also get in touch if you would like to contribute to this series, or let me know if there is a particular study or a researcher that you like me to cover. With this, it is my great pleasure to host Dr. Bruce McLachlan from Cardiff University. Bruce has a research interest in using structural biology, molecular and cellular immunology to study key interactions in immune cell function. He has studied receptors from natural killer cells, CD8 positive T-cells and B-cells, but found his research niche in CD4 helper T-cells, which are obviously a central component of our immunity, but they are often underappreciated and understudied. After completing his PhD at Cardiff University in 2016, he joined the lab of Professor Jamie Rossjohn at Monash University, Australia, where he studied immune receptors of natural killer cells. He then returned to Cardiff back in 2020 to study how SARS-CoV-2 variants evade CD4 T-cell response. Supported by a fellowship from Health and Care Research Wales, his current work builds on our structural and functional understanding of HLA class II presentation to enhance CD4 T-cell recognition of tumor antigens. His overall aim is to develop the foundations for equitable, off-the-shelf immunotherapies for cancer that can stimulate CD4 T-cell immunity across diverse HLA background. That is a great pleasure to have you on board, Bruce, and if you're ready, the stage is yours.
Well, thank you very much for the intro, Hashem, and for the intro and for the invitation to speak today. And thanks everyone for joining. So as Hashem said, I'm a research associate in Cardiff in the Division of Infection and Immunity. And I've titled my talk, what constitutes a CD4 T-cell epitope? And I'm going to hopefully go through what we understand about HLA class II presented antigens. So, why CD4 T-cells? Well, they are these central helpers of cellular and humoral immunity. And, my interest is mainly in the anti-tumor response. And we know that if you can generate a good CD4 T-cell help, you generate a more immune stimulatory environment in the tumor. And these CD4 T-cells can go on to allow CD8 positive T-cells, these killer cells, to go on and eliminate cancer cells. So you need a good CD4 T-cell response to initiate a good CD8 anti-cancer response. And we know that without them, you have these immune cold tumors. And those CD8 T-cells are dysfunctional. They show more markers of regulation and of exhaustion. So, we really need to make sure that we develop therapies that generate an anti-cancer CD4 T-cell response. And Hashem mentioned that I could say CD4 T-cells are understudied, and this slide hopefully tries to demonstrate that. So we have these, seminal meta-analyses that describe just how important CD4 T-cells are in the anti-cancer response. In this one here in 2019, so I put my laser pointer on, they show that, the Th1, these T helper 1 cells, are highly correlative of an effect of a good prognosis in these cancers. And the same is true in this study in 2015. These CD4 memory T cells are up there, the number 2 most important cell type, just behind these gamma delta T cells that are important for anti-tumor immunity in these solid tumors. But yeah, if we look just simply at the amount of studies, papers that are published in the CD4 and cancer space, they get dwarfed by the study on CD8 T cells. So just by way of introduction of this question we have around what do CD4 T cells recognize? Well, they recognize antigens presented on HLA class II. So on the left-hand side of this here is all about antigen presentation. And on the right-hand side is all about how those antigens are recognized by the T cells. It's this important, this event that happens that's, in my opinion, the most beautiful, system within biology that allows these T cells to go off and stimulate antibodies, release cytokines, and then have all these functions to turn on CD8 T cells. So starting with antigen presentation, essentially what happens, the basics are that you have, in case of a antiviral response, you have a virus, which is made up of multiple viral proteins. These proteins get digested up into peptides, which are presented then on HLA class 2 as lengths of around 12 to 20 amino acids in length. And the same is constantly happening in all of our healthy cells. We're constantly processing and presenting these self-peptides that are derived from the self-proteome. And then tumors and cancer kind of sit somewhere in the middle, because these tumor proteins, they can be self-derived, so they are self peptides and proteins. And what's important that all of the rules that whether or not they comes from a self protein or a tumor protein or a viral protein, the rules about which of those peptides that are digested can then go and be presented to T cells is all determined by the HLA. So I'm interested in which of these peptides get presented on HLA, because not all of them are. So maybe around 100,000 of these different peptide sequences make it onto the surface of these HLA molecules. So which of these are, and what are the rules that are governing this process? And in the basics of what these rules are, is all about the biochemical compatibility of a given peptide to bind into the HLA peptide binding groove. So HLA class 2 molecules have these pockets called the peptide binding pockets. And you can see this particularly deep one here at position P1. And it's whether or not a given peptide can bind into this groove is all about the biochemical compatibility of that peptide to sit and be complexed within this binding domain. So in summary, it doesn't matter where these peptides come from. All of the rules are always the same. It's all about whether or not the peptide can fit into the HLA binding groove. And then if it does, we on the other side of the coin, part of antigen recognition, we have this highly diverse repertoire of T cells, each with a unique T cell receptor. Each T cell receptor is generated through these recombination events and has a unique specificity for different peptide antigens. And there's estimates maybe around 10 to the power 8 different TCRs that roam around the body looking for these antigens. And importantly, these T cells and these TCRs are trained to not recognize self too strongly through the thymic selection. But then what comes out of the thymus is a repertoire of T cells and TCRs that are trained not to react too strongly to self and be more strongly reactive to non-self, such as viruses. So, where we are hoping to impact this is to use our sort of cellular and molecular immunology in combination with structural biology to try and decipher what are the rules that govern CD4 T-cell recognition of these antigens. And we can look in donors, we can look in patients. These could be cancer patients, or they could be people vaccinated with viral vaccines. We can look in the blood, isolate the PBMCs and isolate their T cells and try and work out what their T cells are recognizing through techniques such as interferon gamma ELISpots as our workhorse in the lab. And then we have our structural biology toolkit. So we can make these proteins, we can make the HLA class 2 molecules, we can make T cell receptors that see these antigens. We make them in the lab, mostly in E. coli, but also in human expression systems. Hopefully we can crystallize these proteins, which we take to diamond light source, which we shoot with X-rays to work out their structure. So hopefully we can connect the molecular structure and the atoms that derive these functions and these immune responses that we see in donors. And then on top of that, we have a few other important techniques. We have an assay which allows us to measure the affinity or the binding between a given peptide and a given HLA molecule. So these are these HLA peptide binding assays. We can also measure the interactions between proteins. So we can measure the interaction between HLA molecules and T cell receptors. And what we're hoping to also do is have these, computational toolkit where we can do experiments at a larger scale than we can do experimentally. And this is something that I'll come on to at the end. But of course, what is, the most exciting part is using our X-ray crystallography to describe this really important interaction that's happening between a T cell receptor, this one is shown here in blue, and its cognate peptide presented by HLA class 2, shown in sort of purple and pink here. And then in between, in the middle of all this, is the peptide that's been, in this case, processed and presented out of a virus. And then this picture here on the right is the bird's eye view of what a T-cell receptor is, scanning and looking at and deciding whether or not to generate an immune response to. And this image picks out two features of MHC class 2 presented peptides that I'm going to sort of describe in a bit more detail. And that is this core epitope region that's shown here in green, and then these peptide flanking residues that sit either side of the core region that are in pink. And I'll come on to describe why we think these are so important. So my outline of my talk. So I'm going to hopefully by the end of it convince you that we, first of all, don't have a complete understanding of what constitutes a CD4 T-cell epitope. The information that we do have it represents a very poor understanding across the HLA class 2 presentation system as a whole, and that our current understanding is not all that relevant to the world population at all. And then at the very end, I'm going to sort of demonstrate and look at some preliminary experiments that kind of show just how good are we at predicting these peptide HLA class 2 structures, and suggesting that our available data sets are not yet suitable for acting as training or templates for machine learning to predict across the entire system. So first of all, what constitutes a CD4 T-cell epitope? So again, this is the fundamentals. Thinking about comparing to MHC class 1, on the left here, we have an MHC class 1 molecule presenting a peptide. And the key feature about MHC class 1 is it has these pegged down regions at the N and C terminus of a peptide that kind of holds the peptide in. The peptides are typically shorter, around 8 to 11, but up to 13 amino acids in length. But importantly, as these peptides get any longer, they begin to form these bulges in the middle of the peptide. And that's because they're kind of pinned down at these endpoints. But the big difference about MHC class 2 is it has this open-ended peptide binding groove. And what this means is that, first of all, that MHC class 2 presents longer peptides in general. But as peptides get longer, rather than bulging in the middle, they extend outwards. So I kind of like to think of them as a hot dog in a bun, where the hot dog is a little bit too big for the bun. And then these peptide flanking residues shown here in pink are the bits that are sort of hanging out of the peptide binding groove. And MHC class 2 epitopes are not single peptides, they're actually nested sets of peptides. So a given epitope, or core sequence as we would call it, actually exists on the cell surface as a distribution of differentially digested peptides of different abundances. So we can see here there's some data that goes back to 2002 where they eluted peptides off the surface of MHC class 2 molecules. And you can see this is the set of peptides that are actually presented from this one epitope. So in this sense, very different to MHC class 1. But what is generally the same, and the fundamental rules are the same here, is that TCR recognition part. So on the left, we have an example of a HLA class 1 restricted T-cell receptor binding to its antigen. And, it has this well-described diagonal docking angle and quite conserved region and way in which it binds over the surface. And this is practically the same in MHC class 2. So TCRs also bind with this diagonal binding angle, where we have our TCR alpha chain, which sort of hangs out across the beta chain of the MHC class 2 molecule, and our TCR beta chain over of the alpha chain. And importantly here in this purple and blue, these are the CDR3 loops, which are the most variable part of the T-cell receptor, and they hang out, just like in class 1, they hang out most predominantly over the peptide. And this helps to generate the masses of different specificities by the TCR repertoire. So in a little bit more detail. So I'm going to sort of bring you now into some examples of studies that we've done in Cardiff, where we've used structural biology to define what a CD4 T-cell epitope is, or a HLA class 2 presented ligand. So the first one, this was actually work from my PhD here in Cardiff, where we were interested in CD4 T-cell responses to this tumour antigen called 5T4. So as part of that study, we were able to solve the structure of HLA-DR1, which is HLA-DR is one of the isoforms of HLA class 2 that presents antigens to CD4 T cells. And we were able to use the structure to do many things, but the first thing I want to point out here is these features of a HLA class 2 presented epitope. In the middle, we have this core region, which is 9 amino acids and long. And we have these downward facing positions called P1, P4, P6, and P9 that is kind of anchor the peptide into the HLA binding groove. So this, again, we call the core. And then anything to the N-terminus of that, we call the N-terminal peptide flanking residues, and to the C-terminus, the C-terminal peptide flanking residues. And the first thing you can see is just that in the static image, you can see how pinned down the sort of the middle of this peptide is. But then as we go to the termini, it's almost as if the HLA has lost control of the peptide a little bit. And actually what we found in the C-terminus was this thing hooked around and actually started to look like its native whole antigen. And we did these molecular dynamics simulations with Athanasios Papakyriakou, who was at the University of Southampton at the time. And this kind of just shows how dynamic these peptide flanking residues potentially are. You know, the dynamic simulations show the core is kind of pegged down quite well. And then you see the PFRs just exploring the space around. So you kind of see this and you think, well, are these PFRs important to TCR recognition and generating T-cell responses? And the first thing I say is yes, they are, because if you get rid of them, you can no longer stimulate T cells. So this is an example out of Oliver Schofield's work, who's a PhD student who's just writing up his thesis at the moment. And then this top right graphs, these are CD4 T-cell clone that recognizes an antigen. And as Ollie chopped away residues from the peptide flanking residues, the T-cell no longer released interferon gamma in these T-cell activation atoms. So yes, you cannot just stimulate a T-cell with just this core 9-mer region, the pinned down a bit here. And in fact, what he showed was as you add more and more bits onto the peptide flanking residues, you eventually reach the maximal response. So, you know, a HLA class 2 presented CD4 T-cell epitope is not a 9-mer. We say it is, you know, it's at least a 13-mer, which is here in yellow, but really you need these 15 amino acids before you start to get a maximal response. And this is just shown here where he individually removed the N and C terminal PFRs individually. So they need to be there in order to generate immune response, but does it matter what is there? You know, you might think that because these are very dynamic, maybe it doesn't really matter what amino acids are there. And this data shows that actually that the peptide flanking residues do confer some amount of specificity, weak specificity. So in these experiments, Ollie has, what he's done here in the black is taken the fixed core sequence and then put random amino acids within the peptide flanking residues. But this is not as good at stimulating T cells as the native sequence here in blue is. So the further this gets to the left, the more potent the ligand is in these T cell activation assays. And he did this with 3 clones and showed that these random residues don't activate T cells as well as their, you know, the peptide that has been that the T cell has been raised against. So there is some amount of specificity here. And this is just shown here as the contrast to what you see to the core. So, you know, this is an alanine scan at each position 1 to 9 through the core region. And if you mutate, you know, these residues to an alanine, you can completely abrogate response apart from only 2 positions within that core. So really, you know, the specificity of this T cell clone is really hyper-focused on the amino acids that is incorporated in this epitope here. And we're particularly interested in this because we think that the peptide flanking residues offer an opportunity to try and tune and modulate immune responses, T-cell responses in their recognition of these types of antigens. And that is demonstrated here. So this is work that I've done with Professor Andy Godkin and Arwen Gallimore over the years, where we've been able to, you know, bring the structural biology to describe what has been happening in the whole animal. And here, this is an example that was published last year in Cell Reports, where we were interested in generating better influenza vaccines. So this is a murine model of vaccination of influenza, where the mice are vaccinated with a whole virus, X31, which has been modified to include a single modification to that virus, to include an arginine at this position within the C-terminal peptide flanking residue that our previous work showed was able to generate more avid T-cell responses. And when these animals were vaccinated with this 11R variant, they were then better protected against re-challenge of a heterotypic influenza vaccine later in time. So these modifications, whilst it's just a single amino acid difference in what people might think to be an unimportant part of the epitope, actually had a whole animal difference in the protection of these animals. And, you know, we could come in and we can see that when we add this arginine here at position 11 to the X-ray crystal structure, that we just, in general, we get these more bonds forming, a more interesting peptide flanking residue, maybe creates more interactions with the T-cell receptor. And this is enough to get over some sort of activation thresholds and generate a more functionally avid T-cell response. Oops. The next thing I want to point out is just how difficult this can be without the structural biology. So again, on the left, so here in this study in 2019, we had T cells that we knew recognized this 20 amino acid long peptide, but we didn't know where the core was. So, this isn't necessarily a very strong motif for HLA-DR1. So without doing the structural biology, it was difficult to, define where this core region was. Was it here, 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 here? And this on the right here is from a preprint that we've released this year, where in the context of an anti-tumor response, we weren't able to do the structural biology, and the amount of work that went in to try and define and find these minimal epitopes based on the reactivity of these CD4 T cells was actually quite difficult. We had to get these truncated peptides where we knock it off and make them shorter until the T cells no longer respond to it. And the other thing to point out is that we were actually led down blind alleys here, because the predictions of what this core for this epitope was actually not what we found to be true. So it's a difficult thing to predict as well computationally sometimes. But it's a very important feature of a CD4 T-cell epitope. And again, this brings in work that we did during the pandemic. It's published in Cell Reports, where we were interested in defining epitopes of CD4 T cells against SARS-CoV-2, the causative virus of COVID-19 disease. And during the pandemic, we were screening and trying to look for immunogenic epitopes, and we found a number of them that we were able to describe again through structural biology. But what was really interesting was that when the Omicron wave came along, and the SARS-CoV-2 virus incurred quite a few mutations, a couple of our epitopes we were interested in became mutated, and this is one of them here. So this epitope was interesting, and I want to point it out because, again, what we saw in the structural biology couldn't have been predicted. We found one amino acid that was mutated in the Omicron variant, and this was this asparagine, which in the wild type, or the Wuhan HU1 ancestral strain, was an asparagine. And then when Omicron came along, it mutated to have not an asparagine at this position, but a lysine. And what actually happened was not predicted by both NetMHC and ourselves looking and predicting what might have happened. What happened was that this lysine was no longer a favorable, had favorable biochemical compatibility for the P1 pocket. And in fact, then the peptide was presented in a completely different way through this register shift. So what does this mean? Well, it just shows just how a single amino acid change can have such a drastic change on what a T-cell epitope looks like to a T-cell and a T-cell receptor, and just how finely poised the system is. So we know that T-cell receptors are highly cross-reactive. They can bind, millions, maybe more than millions of different peptide sequences. But if the HLA presentation is so finely tuned, this is not going to be picked up, highly unlikely to be picked up by cross-reactivity of a T-cell receptor. So I just want to demonstrate how important the register is in defining an epitope. So in summary so far, I've said a T-cell epitope, a CD4 T-cell epitope is a 15-mer peptide, maybe a 13-mer peptide, but a 15-mer peptide seems to be optimal, not a 9 amino acid sequence. But probably the epitope core seems to be driving the majority of TCR specificity, and we can see that when we start to try and mutate and change the core. Whereas these peptide flanking residues, they sort of maybe are very mobile, and they seem to confer weak TCR specificity, but we think that they present an opportunity to try and tune TCR recognition. Defining what the core register of an immunogenic peptide can be challenging, but it's really important because that is the first key feature of a HLA class 2 presented epitope. So next, I want to convince you that overall, we have a poor understanding of HLA class 2 presentation when we consider the variation and the diversity across the system. So the first thing to point out is that we don't have just one family of HLA class 2 molecules that present antigens to T cells. We have 3 isoforms, HLA-DR, HLA-DQ, and HLA-DP. And within these gene sets, we have incredible amount of polymorphisms and genetic variants. The most genetic variants sit on the HLA DR beta chains, where in the IMGT, which collates these alleles, there's over 3,000 of them. And I'm going to show this image again later on, but all I want you to see here is that in each of the most populous countries in the world, the most frequent HLA DRB chain, or the most important allele, changes in each of these different countries. And of course, these polymorphisms occur most frequently in the regions that are important for binding peptides. So these circles here represents the polymorphic residues. The bigger the sphere gets, the more different polymorphisms have been observed within just this set of 26 alleles. And we can see then how that influences the peptides that are bound by these different allotypes. So we have an incredible amount of variation in the level and diversity on the level of the HLA class 2 genes and alleles. And then we have an incredible amount of diversity in the number of different peptides that each of these alleles can present to CD4 T cells. So, we can see that, these alleles, which have these polymorphisms that I've just shown around the peptide binding groove, we have these allele codes that help us to track the names and these different variants. But importantly, these different variants that cause different peptide motifs and preferences for different peptides to be presented by different alleles. So if we think about this and contextualize it, we have different people around the world who have incredibly polymorphic HLA molecules, and in each of those different individuals, they present potentially different peptides from SARS-CoV-2, or different peptides from different tumor antigens. And to be able to work this all out is incredibly challenging, to be able to capture all of this diversity. And sort of the way I've been thinking about it is, to try and minimize all of this diversity down into this grid here. So on the x-axis, we have the diversity of all the different HLA allotypes, and I'm focusing here on HLA-DR. So from this point here, we have a HLA-DR allotype that is most different from the one that's over this side. And on the y-axis, we can think about all the different peptides that each of these different allotypes present on their surface. Down the top, we have the strongest binding, and then down the bottom, we have the weakest. And just to demonstrate here, illustrated from C to D, these two alleles have one single polymorphism difference, but then these polymorphisms will change the preferences of these different peptides. Again, relating back to the people, meaning that different people will present and have immune responses to different peptides in the body. So in order to try and capture the depth of understanding we have across this system, I went through an exercise of, scouring the PDB to try and understand the structural state of play for MC class 2 biology. The method was to use a set of sequences that use the RCSB search API, and then that captures a big library of potential HLA class 2 containing structures, and then they're blasted against the IMGT HLA database, which is sort of the gold standard for all of these different HLA alleles that have been observed across different populations. And in doing this is what I see. So, the amount of deposition. So if one deposition describes a distinct peptide HLA class 2 complex, we currently have, we're dwarfed in the number of these structures we have and the amount of data we have compared to MEC class 1 biology. We have 5 times more depositions that are containing MC class 1 molecules. And if you look at the progression of these over time, the number that we currently have today of MC class 2, 274, was achieved 16 years ago for MC class 1 biology. And the same is mirrored if we look only at human depositions. We have 5 times more depositions of HLA class 1 than HLA class 2. And then if we start to define what different organisms we've described across the animal kingdom, MC class 1, we have these descriptions that extend across the animal kingdom from humans down to sharks, many different organisms, whereas in class 2, we have human, mouse, chicken, and recently bat. And interestingly, we have more depositions that describe mouse or murine MSC class 1 than we do human MSC class 2 descriptions. But we do have, there's a lot of interest in biology around MSC class 2 molecules. And if we try and capture, within this data set of 207 structures, how many of them describe just peptide MHC structures alone, and how many of them are bound to other things. So, I find 64 peptide HLA structures too that are bound to T-cell receptors. And from this, you can begin to see the patterns of binding. So you can see this overall conserved topology where the alpha and the beta chain are loosely overlapping on each other, apart from these few outliers which describe T cells that don't necessarily cause TCR triggering, which binds outside of the norm. So we have this sort of, set of structures that are contained within the PDB. So the next thing I want to do is just kind of give a flavor of how much of the diversity of the system that these structures describe. And if you go back to Christopher Thorpe's talk, he gave an excellent kind of overview of this, and also in the context of MC Class 1, and he is able to put these numbers into a lot better context than I can. So I'd encourage you to go back on the YouTube and watch Christopher Thorpe's webinar if you're interested. So again, we have DR, we have HLA-DQ and HLA-DP. Most of the depositions in the PDB are of HLA-DR. Within HLA-DR, we actually have 3 or 4 genes that encode DR beta chains. The majority of structures are from this DRB1 gene, which is the most polymorphic and is present in all people. And for that reason, that's why I'll focus mainly on HLA-DR here. And by HLA-DR, I mean alleles of the DRB1 beta chain. Hopefully that makes sense. So consider the variation of HLA-DR beta chains. Importantly, in HLA-DR, the polymorphisms are only generated by the DR beta chain, and that helps because it's a much more reduced system. So we don't have, pairing between variant alpha and beta chains. We have a much simpler system to study here. There are, nearly 2,500 genetic variants of DRB1. 86 of these variants have been deemed common and found in multiple populations, but we've only described 12 of these. And that's shown by this graph here. And probably more significantly is, of those 12, we have considerably more data stacked towards only 2 allotypes of HLA-DR, and that is denoted by the codes 0101 and 0401. So these are called DR1 and DR4. So we have, although we have this massive variation, it's all stacked towards these 2 alleles that dominate the data sets that we have solved so far. And this is trying to show the coverage across these polymorphisms. So again, this is just showing 26 different alleles. And the sequence at the top here is the sequence of HLA-DR1 as a reference allele. And then below that is the occurrence of any polymorphisms that occur within these only 26 alleles. And in gray, these are the polymorphisms that we're yet to describe structurally. So these are what we still don't understand, how these polymorphisms influence the motifs and the preferences of these different alleles to bind peptides. So, if I try to minimize it down to this whole system, again, I said we've got this massive variation that's contained within this grid. We have 274 structures that describe MC class 2. 207 of them are of human MC class 2 molecules. Some of them don't have complete peptides. They're incomplete and they have things like ligands, small molecules bound in them. If you consider the amount of redundancy that's in those 197, there are 115 unique peptide HLA pairings, and this includes peptides that are citrulinated. If you remove those citrulinated peptides, we have 96, truly peptides that are presented by HLA class 2 across the whole system. So, on this axis, we have a diversity of thousands, and on this axis, we have the diversity of hundreds of thousands. And this is all described by just 96 peptides. Sorry, 96 pairings. So, so I said, we don't have a very broad understanding of these molecules, but how relevant is the current understanding and of the structure we have solved to the world population? So I'll bring this back up again here. We have the most, 7 most populous countries in the world, which accounts for around 50% of the world population today. And in each of those different countries, each has the most common and most frequent allele of HLA-DR beta chains is different in each country, apart from the sharing of this allele 0701, which is shared between India and Brazil. So, if you're in America, the most important allele to Americans in general is this 1501 variant, whereas in, some of the most populous countries, India and China, with a, very large number of people, we have these other alleles that are important to these different people. And one way you can look at this is, rather than look at the country level, we have these really well curated data sets that come from the stem cell registries. In the UK, the registry is maintained by the Anthony Nolan charity, and I'm going to focus on a data set that they published that describes what allelic variants of HLA-DR beta chains are important to different broad ethnicities in these people that are on the registers to be potential stem cell donors. And I have to say, this paper that was published in 2020, Leon et al., is probably the one that I keep going back to the most, because it really demonstrates just how poorly the data we have today is relevant to different people around the world and different people of broad ethnicities. So what is shown here is the 10 different broad ethnicities that are defined by the Anthony Nolan registry. So when a person joins the registry, they tick some boxes to self-declare their ethnicity. And then Anthony Nolan has grouped these into broad ethnicities. And this has shown the top 10 most frequent alleles of HLA-DR in these different broad ethnicities. And then at the end of the bars is the number of structural depositions we have for each of these alleles or allotypes. And this group here, BINWE, it stands for British and Irish Northwestern European. And we can see that if we summate all of those structures we have a massive relevance to these people. But if you are of African or Caribbean ethnicity, the structures that we have today do not really represent the alleles that are important to these different people. And hopefully you can see that here in these graphs. So if we were to study one single allele, which one should we be studying? And the eagle-eyed in the audience might see that, in my opinion, we should be studying HLA-DR7 or 0701, because it is the most broadly relevant allele and the most highly shared between each of these diverse ethnicities. And this then expands this on to look at three registries. So the one on the left is the Anthony-Nolan data set. It's the same data set from the previous slide. And now we're just plotting the allelic frequency of DR7, which is in pink, or 0701, compared to 0101 and 0401, which is where we have all the data today. And what you can see is in all of these broad ethnicities in these data sets, DR0701 is far more relevant to these people than 0101 and 0401, where we currently have all the data focused. And you can try and do this at a country level, but the data is a little bit more patchy. But what you can just look at here is at the left-hand side. So this is ordered by population size. We have some of the most populous countries in the world. And in all of these, DR0701 is more important to these people than the alleles that we've studied in detail. So DR0701, what do we know about it? We have a single structure that describes not an antigenic peptide, but describes how the CLIP peptide, that is this placeholder peptide that is put in the groove when the HLA-DR is made. So we don't truly understand why this really important allele presents the peptides it does. And that is what we want to try and improve on. So lastly, I want to just go through, how good are we today at predicting these structures? So it takes a lot of work to solve these structures experimentally. So can we make predictions that will allow us to, study this on a scale that we can't achieve in the lab? And I want you to see me more as an end user of these models, because I don't truly understand how they all work, but, I can run them. So, I want to then know, do these models actually tell us anything about the biology, and can I use these to infer anything interesting about HLA class 2 biology? And why do I think this might be important? Well, if you think about the scale of things, could we simulate what is happening in the thymus if we could accurately model every peptide HLA class 2 complex that is presented in the thymus to our T cells? Could we simulate thymic selection through computationally predicting the structure of all of these potential ligands? And that's why I think, the scale is important here, and being able to study this at a different scale that we can do experimentally through experimental structure determination. So again, I'm going to bring back this grid. And again, I said we have the diversity of this system. And, speaking to the people who do, I kind of understand how these ML models work. The best data set to train these models on would be one that has experimental templates or, training data that is spread out across the entirety of the system. And this is my best attempt at trying to show just how poorly those different structures map across the diversity of the system. So you can see DR1 and DR4, where we have all the structures. We do have quite good depth here. We have descriptions of how strong binding peptides bind, and we have descriptions of how weak ones do. But this distribution is not across the entirety of the system. And, I think if we are to try and decode or unravel CD4 T-cell recognition through some sort of structural prediction approach, it's important not to forget about the HLA, because that is the first thing we need to be able to accurately predict. There's no point in predicting T-cell receptor structure if we can't predict what the antigen looks like. So this is my sort of attempt at benchmarking and trying to work out, how good do I think, or how good can we see that these prediction models are at predicting structure. So I've used 3 different models so far. One's called T-fold, which is based on AlphaFold 2. The next one is AlphaFold 3, and this is running just normal AlphaFold 3 through the server. And this one, T-fold TCR, which emerged in 2025, that comes from the Tencent AI lab. Unfortunately, we have 2 models that are named very similar. We have T-fold and T-fold TCR. But the thing to point out is that T-fold TCR can also predict the antigen through this PMEC module. So, we're constantly solving these structures of peptide HLA molecules. They are not yet published, so they're not yet in the PDB, and they're not yet able to train machine learning. So they represent a good opportunity to try and benchmark how good these are at predicting what we can see in the lab. And I don't want to sort of go into the details of each of these structures, but I just want to point out, broad brushstrokes about the biology here. So if we look across the top at T-folds, we can see that it is actually quite well predicting, at least 4 of the 5 conformations of the core region of these epitopes. So in yellow, we have the experimental unpublished structure, and in green is prediction. But AlphaFold 3, so AlphaFold 3 is trained on structures that include shorter peptides. So potentially AlphaFold 3 might be better at predicting these kind of complexes. And we can see it does get a few of them broadly right. But we start to see these, helices starting to come in. And, it's not, as we'll see, it's not perfectly predicting the biology. And then this T-fold TCR one, at least in these examples, the thing that it doesn't seem to be quite capturing is the register. So if we just summarize this all and just look, do these different models correctly predict the core register? T-fold is getting 4 out of 5 right. AlphaFold 3 is 3 out of 5. And T-fold TCR is maybe getting 2 of them right. And I say maybe because it kind of starts in the right place, but then it actually starts to get lost in the groove. And that's something I'll show in a second. So why do I think the T-fold is doing a better job? Well, it includes a module that predicts register in this part here. And also what it's doing, it finds a closest template, and it uses that as a template to model against. And I think that kind of constrains it to what is the more observed biology of these complexes. And just out of interest, if I've added this down the bottom here, NetMEC2 is correctly able to predict the core of these, each of these different epitope or ligands. So, whilst it doesn't predict the structure of them, NetMEC2 in these examples can accurately predict the register. And I do note that this preprint came out a couple of weeks ago, and I was hoping to do some predictions with this in time, but I haven't quite made it in time yet. But importantly, this pipeline here incorporates NetMEC as part of its prediction pipeline. And to me, this seems like a sensible approach. So if we were to try and make these predictions en masse, this is a lot more difficult because we don't have these kind of experimental versus predicted examples to benchmark against. So what I've been doing is trying to make a test set for, demonstrating an en masse approach to HLA-DR prediction across different allotypes, again, on this axis here, presenting different peptides, being the strongest binders to the weakest binders. So what I have is a set of 22 HLA-DR allotypes, 12 of which are those that have been solved experimentally, and then 10 unsolved, but what I deem important allotypes to different people. And then on the y-axis, what I've done is taken the human proteome and used mixed MHC2 pred, which is similar to NetMHC that you might have heard of, and predicting with each of those allotypes the best binding peptide within the human proteome as a reference sort of set, down to weaker binders at approximately 50% rank EL. So what we have then is a test set of 440 pairings of peptide HLA-DR to try and predict. And the first thing I want to point out again is this idea around predicting the core. And what is seen in running these models, the uncertainty in predicting the core. So Tfold produces up to 34 different models I've seen in this data set. And then what I'm looking at is within those 34 models, does it always predict the exact same core? Because I think that infers some amount of confidence in its prediction. And then the same with AlphaFold 3. So AlphaFold 3, at least in my hands, spits out 5 models on the server. Within those 5 models, how many different cores does it predict? And I call this core uncertainty. And then so this is the output of that analysis. So I'll go through it again slowly. In this one here, so this is Tfold, which is the AlphaFold 2 based model. On the left-hand side of this grid, these are the HLA-DR allotypes that have been solved experimentally, and they're actually ordered by the number of structures. So this one column here is DR1, and this column here is DR4. And then from top to bottom is predicted strongest binder in the human proteome down to weaker binders. On the left, AlphaFold Tfold, in the middle, AlphaFold 3, and on the right, Tfold TCR, at least in my hands, only produces one model. And that seems to be true even if I give it different random seed. So we can't really look at Tfold TCR for this. But what you'll see on the left is the uncertainty. So the more yellow, meaning the more different cores that's predicting across its models, increases as it gets to weaker peptides. And I think that makes sense because the motifs are probably not quite as strong. But actually, if we look, and this black line here separates the solved allotypes from the unsolved allotypes, where there is no experimental structure, the ML is worse and more uncertain about predicting even strong binding peptides. So this to me suggests that we need more data of these important unsolved alleles to these people. That is replicated in AlphaFold 3 as well. So if we look at the top right-hand corner, this is the best binding peptide, theoretical binding peptide in the human proteome, and AlphaFold really doesn't know what its core looks like for this allotype here that is not solved. So the next thing I've looked at is the orientation of the peptide that's binding. So some of you may know that the majority, and at least for HLA-DR, all of the peptides presented by HLA-DR have this forward or canonical orientation, where the N terminus is at the P1 and the C terminus is at the P9. And there's examples in HLA-DP where this can reverse, and this is described here by this paper. But there's no evidence so far that really suggests that these reverse binders occur in HLA-DR. And what we can see is that Tfold actually fixes it in a forward orientation. So all of these are always in forward, and that's a good thing for HLA-DR, but maybe a bad thing for HLA-DP. But particularly this Tfold TCR is predicting even some of these really strong bindings actually bind backwards. And I don't think that you could argue this is predicting, new biology, but everything that we've solved so far experimentally points towards this being highly unlikely. And this is also replicated when we look at whether or not the peptides stay in the groove. So what I've looked at here is whether across the P1 to P9, whether the peptide begins to move away and deviate more than 3 angstroms away from what is the norm. And this is an example of one of these predictions here, where the peptide is essentially coming out of the groove. And Tfold TCR seems to predict quite a lot of these. And again, you could say that this is predicting biology, but all of our data, experimental data sets say that the peptide is pegged down across the core region. So in summary of this section, I think peptide HLA class 2 structure prediction is quite a unique modeling problem, and that register prediction is kind of that first big but critical challenge that the models need to be able to learn. I think there's something in this template modeling. Tfold uses a template, it finds the nearest template, and then I believe it models upon this. And I think this helps it to conform to the known biology. And for me, this is, I think, why this Tfold, which is based on AlphaFold 2 by Mikhailov et al., seems to produce the most sensible predictions. I think there's something in the performance and the confidence at predicting our solved allotypes. And I think that's important when we consider how relevant our solved allotypes are to the world population. And I think we need to be better at assessing, the biological sense of these HLA class 2 predictions, rather than just, it's quite hard to do these experimental versus predictions because we have very few examples. But we need to look at the outputs of these models and actually think, do they actually make any biological sense? So in summary, I hope I've demonstrated that, the important features of HLA class 2 presented antigens, these longer peptides. I've shown how our understanding of HLA class 2 is, behind the MHC class 1 field, 16 years behind, and that what we do understand about HLA class 2 is significantly skewed towards European-centric allotypes of HLA-DR. And this may drive poor predictive ability by ML structure prediction models that may have issues and may generate inequities going forward as we try to incorporate these kind of predictions into generating therapies. So that leads me to thank everyone who's been involved in the work. So particularly Ollie, who's currently writing up his PhDs, who I co-supervise, particularly Andy Godkin and Arwen Gallimore, who are my PIs and my mentors across the years. Pierre Rizkala, who's now retired, who taught me, when I was a PhD student about crystallography. Collaborators who have been involved in the work, and most of this was funded by Health and Care Research Wales Fellowship. And the SARS work was funded by the Welsh Government. I showed some work from my PhD that's funded by Cancer Research UK, who are now also funding our project to try and use what we've learned about these epitopes to try and generate better anti-cancer immune responses. So thank you all for listening, and happy to take any questions.
Thank you so much, Bruce. It was really fascinating work, and it was very well pitched and very well presented, very clear. Thank you. We really enjoyed that. As people are thinking about questions, I would like to take 1 minute of your time for two announcements. One is that obviously this session was the last session of this year. We usually have our webinars first Monday of each month, but for January, it will be 12th of January. And I will have a great speaker from University of Pennsylvania. So please stay with us, and I'll be delighted to welcome you for that seminar. So the very first question from Bruno Raposo. Hi, would it be possible to get access to recording? Oh, that is a question for me. Yes. Okay. Actually, I have a number of questions, but I'll go with one. I found, from systems biology point of view, this your observation about this flanking area very, very interesting because, it helps you to, incorporate new features for enhanced peptide immunogenicity and so on and so forth. And also, your findings and observations from how to incorporate this structural information. So my question is that to what extent these rules are generalizable? I mean, in your work, you were mostly looking into, I think it was just one HLA type, DR1. And also, how is it for instance, T helper cells versus regulatory T cells? Is there any example of those?
Okay, so how generalizable is that something that we're working on? So we as part of the program going forward, we want to try and work out whether it does generalize across more allotypes and more important allotypes to cover more people. We've done this all in HLA-DR1 because experimentally it's what seems to work for us most easily. But now the hard work is to try and show it in multiple different allotypes so that it's applicable to more people. But what I would say is that in terms of generalizability, we seem to see that a similar molecular features that if we add to these peptide flanking residues seems to increase TCR affinity and immunogenicity. And that seems to be, these positive charges or these large, bulky amino acids. So it seems to be some amount of generalizability in that sense, but we need to show it in different HLAs. And then the second part of your question was Tregs versus T helper cells. So that's where the animal models come in. In the animal models, when we made these changes, we didn't see that there was a change in phenotype to grow out any more Treg cells. And again, we're exploring that in a bit more detail by pairing TCR repertoire analysis with single-cell RNA-seq to try and make sure and identify whether we induce any more regulatory features within these mice, and ensuring that these modifications we make do in fact still induce a T helper 1 response.
Excellent, thank you. Awesome. And the next question from Robert Spapen. Thanks for the insightful talk. I am wondering whether instead of modulating the flanking peptide sequence to get improved TCR recognition, do you think you can also improve recognition by changing the TCR sequence? Prediction question mark.
Sure. So, if you look at the work of companies like Immunocore, so they're very interested in doing that and generating these high affinity T-cell receptors that can bind better to antigens. There's not really any examples of them doing this yet for HLA class 2 presented antigens, as far as I know. But I think what we are more interested in is generating vaccines, because I think we see these as being more off the shelf and equitable therapies, than these cellular therapies or, very specific therapies that you can do if you play around with the TCR sequence. But if we can, induce a stronger T-cell response through the antigen, then we can design vaccines that incorporate these changes into the antigens, and then hopefully they can infer better protection. Okay,
fascinating. And another question, the example that you basically presented in case of SARS-CoV-2, how generalizable or how transferable that is, to, for instance, cancers where we have, a higher variability of mutational space.
Yeah, I mean, that I think is something that is a really interesting observation. One single mutation can cause a completely different register, causing a different epitope. When you think about that in the context of cancer, a neoepitope that induces a register shift to create something that the thymus is not seen in the thymus could present a very immunogenic epitope in cancer. How generalizable that is, we don't know, but if we can accurately predict these things, then maybe we can build models based on predictions how this epitope causes a register shift, or a very different epitope which is not seen in the thymus. Does that make it immunogenic? And I think that would be the power of being able to do these kind of predictions at scale.
Thank you. A follow-up question from Robert again, and the question is that, do you have an indication then of how many of your induced TCRs using vaccinations are then not recognizing your presented epitope anymore?
So that is important. So the way we formulate our experiments is that we're only interested in modifications to the flanks that expand T cells or T cell receptors that can still recognize the wild type epitope. And if you go into the preprint that's there, so it's on Research Square. So what we did was a bit of TCR sequencing analysis to see what, when we stimulate T cells with the modified peptides, what TCRs we grow out. And then when we re-stimulate those T cells with the wild types, do we get the same TCRs? And the answer is yes. So what we think we're doing is selecting the same TCRs, but just giving them a little bit of extra hand that causes them to expand. But then we're screening only for modifications that allow those TCRs still to recognize the wild type, and because that would be important for our vaccine model.
Excellent. The next question from UGU, a very interesting talk. Do the PFRs form contacts with TCRs?
So yes, they do. So if you look at some examples, including the the PKY epitope, which is kind of like the best described HLA class 2. The lysine, the K in PKY is a N-terminal peptide flanking residue, and it's really important in binding to the TCR and by TCRs that have been solved structurally. So yes, we do think that they are being bound by the TCRs, and probably the further you get away, probably the less they're contacted. But I do think that these are mobile, and they're maybe going up, they're maybe going down, and making contacts with both.
Excellent. And another important question from Marcin Pikaleski. Since numerous HLA class 2 crystal structures use covalently linked peptides, for example, beta chain linkers, to stabilize the complex for folding and crystallization, how much do these engineered linkers affect current models and predictions?
Yeah, so I've looked at that. I've got that number down somewhere. I just haven't shown it. But that's the reason why. So we don't put any linkers on our peptides that we are interested in. That's why we prefer the refolding technique, where you don't have any linker. And that is because when you look at the structures that have these linkers, they are definitely, the conformation that the peptide flanking residues take is definitely influenced by the linkers. So the ones that are linked to the TCR, they just go up towards the TCR. And the ones that are linked to the beta chain go towards the beta chain. You can see it when you look at these different structures. So yes, they definitely influence the biology. How they influence the predictions and the models, I'm not sure, because I don't really expect the models to be able to predict very well what's happening in the PFRs because they're so flexible.
Fantastic. And that brings me to next question from Sandeep Kumar. Is there any guideline how strong should be the binding? Because it is too strong that TCR clone will be discarded not to have autoimmune reaction.
Yeah, so I mean, I can't off the top of my head know what that number is, but there's models that show that at some point then they become Tregs, and at some point they are deleted. And Treg TCRs are thought to be higher affinity, and that is why they become Tregs. And in terms of in our modification models, we about the peptide flanking residue modifications, we haven't gone too strong yet. And I should, when we talk about making a higher affinity, we're talking about a doubling of the affinity by SPR. So we're looking at kind of bringing those cancer reactive TCRs to be more in line with what a normal antiviral TCR would bind. So it's not like the high affinity matured TCRs that have nanomolar affinities. Just tuning it a little bit to still be within the natural normal range for TCR interactions, but just closer to that low micromolar range of antiviral TCRs.
Fantastic. And the last question, considering PFRs now, based on your work, offers basically potentials for modulating immunotherapies. So how do you envisage about triggering cross-reactivity, T-cell cross-reactivity?
So triggering cross-reactivity, if you mean between our modified and our wild type, is that what you mean? Yeah, I think when we look at how permissible that T-cell clone that I showed is to changes in the peptide flanking residue, it doesn't have to be a particular amino acid. So they clearly are amenable to changes. I think that's a feature of these PFRs that make them amenable to these kind of modifications. So, we assess the cross-reactivity by growing out T cells and testing their ability to recognize wild type and modified, because that's what's important to us. But, I think it's, a feature of these, the weak specificity that these PFRs induce on TCR binding that, allows them to have this level of cross-reactivity to these antigens.
Fantastic. Thank you so much, Bruce, for yourself and your teams and colleagues at Cardiff University. These are really great work, and always good to catch up with you guys. And thank you for the audience that you tune in month after month. That means a world to me. And love to see you next year. And I wish you a Merry Christmas and Happy New Year. Thank you. Bye-bye.
Thank you.